Good morning, good afternoon, good evening to all of you joining us from around the world today. Welcome to the scientific webinar entitled Thermal Physiology, the Effects of Environmental Temperatures on Energy Expenditure in Mice. This webinar has been made possible by Columbus Instruments, so I would like to extend gratitude to them for enabling us to gather today and share science together. Our speakers today are Dr. Marc Reitman, Branch Chief and Senior Investigator from the National Institute of Diabetes and Digestive and Kidney Diseases, and Dr. Oksana Gavrilova, Staff Scientist and Core Director from NIDDK. Together, our speakers will be giving us an in-depth overview of thermal physiology, energy expenditure, and thermoneutrality in a mouse model.
To present this talk on behalf of myself and Dr. Oksana Gavrilova, and I'd really like to thank Inside Scientific and Columbus Instruments for the invitation to do this. So I have a very brief introduction, which is, why should we study energy expenditure and why should we be doing it in mice? And as a scientist, I'm happy with doing science for science's sake, but I think it's important to understand physiology, both in order to inform our medical research and medical care. And mice are the most studied laboratory mammal, so it's a relatively cheap, manipulatable, genetically tractable system that's very, very widely used, doesn't need a lot of drug to test. However, fundamental differences exist in energy expenditure between mice and humans. That's the main theme of today's talk. And in order to take this into account, we have to understand mouse energy physiology to identify conditions where mice might be a better model for human physiology. And again, this will inform anti-obesity drug design and development and increase the probability of success. So you can do fewer clinical trials in people and more work in model systems like mice to be a more efficient and effective drug discovery process. So today's talk will have three parts: a short review of indirect calorimetry, another short introduction to thermal biology, and then the main part of the talk will be multiple vignettes on environmental temperature, body temperature, and energy expenditure. So I just want to share sort of my motivation for studying physiology as it relates to obesity. This is a slide that's actually now from a defunct website that Morgan Downey used to run, where he went through the literature and came up with 104 associations with obesity, putative causes of obesity. And, obesity is so much in the public eye, it's very easy to quantitate, to do association studies. And I want to point out that genetics is only one of these 104 putative causes, and yet genetics is already probably a few hundred, at least, loci that contribute to the variance of adiposity. So I would like to argue when you see a slide like this, it really means we don't know enough about the physiology to be studying it rationally. So my view of obesity physiology is that it has three factors. There's food intake, which will not be talked about much today, energy expenditure, which is the main subject of today's talk, and the third thing is the regulation that matches those two. And I think that's really the underlying cause and interest in obesity. And in order to understand three, you have to be able to measure one and two accurately and reproducibly and precisely and understand what you're doing. So what I'd like to now talk about is energy expenditure. And energy expenditure is typically broken down into four components. There's the basal metabolic rate, which is the energy needed to keep the body running, awake, at rest, in the post-absorptive state, in a thermoneutral environment. Other contributors are the thermic effect of food, which is the amount of energy expended after a meal and is commonly ascribed to the energy needed to assimilate the molecules in the food to molecules stored in the body. There's the energy expenditure required for physical activity, and there's the energy required to warm the body, cold-induced thermogenesis, to allow you to maintain your core body temperature. Now, how do you measure energy expenditure? We're going to talk today about indirect calorimetry. And the basic principle of indirect calorimetry is you're measuring the rate of oxygen consumption, the rate of CO2 production. What you do is you put a mouse in a reasonably small chamber where you pump room air in at a constant rate and pump it out also, and measure the difference in oxygen between in and out and the difference in CO2 between in and out. And this metabolic chamber or metabolic cage is inside a larger thermoregulated chamber that allows you to vary the environmental or ambient temperature. And the mouse is also monitored by infrared beam break for physical activity. And we almost always also measure food intake and core body temperature by telemetry when we do indirect calorimetry. And I just showed you an example of the systems we use from Columbus Instruments. We've used both the home cage. We have both the home cage system and the other Oxymax system. Okay. Now, again, very little background on the basis of indirect calorimetry. What you're doing is you're measuring oxygen consumption, and it turns out that oxygen consumption from carbohydrates about 112 kcals per mole, oxygen consumption from fatty acids 104 kcals per mole. So if you just measured oxygen consumption, you'd be within 5 to 8%, even at the extremes, of the correct metabolic rate. In doing calorimetry in mice, we typically do not consider the metabolic contribution from protein molecules or amino acids. In humans, that is done, but it's relatively constant and a relatively minor component, maybe 10, 15%, and is not typically considered in mice. And I'm not going to talk about it any further today. But in addition to oxygen, you can also measure carbon dioxide. And this actually provides a lot more useful information. And the way we usually express it is the ratio of CO2 produced to oxygen consumed, which is something called the respiratory quotient or the respiratory exchange ratio, RQ or RER. And the point is that the RER when you're burning carbohydrate is 1, whereas the RER for fatty acids depends on the exact fatty acid, but for saturated palmitic acid, it's 0.70. And so you can know that where you are in that scale between 0.70 and 1.0 tells you the fraction of carbohydrate versus fatty acid that you've been oxidizing. And what I thought I'd do is give you some of my biases and thoughts about how to approach and how to report indirect calorimetry results. The first thing is I really like reporting total energy expenditure as, for example, kcals per day per mouse. Reporting the raw oxygen consumption and raw CO2 production rates does not add any information above the total energy expenditure and the RER. However, reporting the RER is really useful. It does tell you the fuel source, but it also is a very important biological technical check. Is the RER biologically plausible? Is it between 0.70 and 1.0? And on a finer scale, the respiratory exchange ratio over long time scales, multi-day experiments, should approximate the food quotient. The food quotient in our chow diet is about 0.91, and the food quotient for our 60% high-fat diet that we've used is 0.79. So I really like the RER because it gives you a chance to make sure there's a diurnal rhythm, that the numbers make sense. And it's a sort of a finicky measure, so it's nice to see it. And finally, the other thing I really like to see in metabolic rate experiments is in long-term experiments, the energy expenditure should equal the food intake. So if you express the food intake in the same units as the energy expenditure, such as kcals per day per mouse, you have a nice technical check that the energy expenditure and food intake are roughly comparable. If they're not, did you make an error in measuring one of them? If not, was the animal unhappy? The animal was in the chamber, not used to the chamber, and wasn't eating. So that brings up another point, which is the calorimetry chamber is a fairly unusual environment. It has air rushing through it. So we, if at all possible, like to do a 2-day acclimation of the mouse to the calorimetry chambers before we do any measurements, so that we don't get funky mismanagements, for example, between the food intake and the energy expenditure. The other point I'd like to make is that energy expenditure changes are really biologically significant, even if very minor. So a 5% change in energy expenditure over time is very biologically significant, but to have a low type 2 error probably requires maybe 100 mice. So realistically, you're more likely to be able to detect a 10% difference in metabolic rate with a 6 to 10 number of a mouse experiment. But you really, the more mice, the better in these sort of experiments, and realize that the precision of the method is going to tell you a lot about the biology. Okay, the only other thing I'd like to talk about methods is how do you express metabolic rate, because this is really done in problematic ways much more often than it's done correctly. And the thing I first want to say, if it is at all possible, study mice groups with the same average body weight and composition. Group A and B should weigh the same thing. If that's true, none of what I'm going to talk about matters. But that's often, and I would also argue that if you do have mice that weigh differently, study them earlier in life where they weigh the same, because that'll also be a valuable readout on the biology of those mice. So what I've done is, this is discussed in detail in a paper that Matthias Chopp is the first author, and the figure here I've taken from a review, from a paper from John Speakman, and the references are given. So on the right-hand side, you see 2 groups of mice that have the same energy expenditure shown in the middle bars. But if you look, actually, the lean body mass, but you could have done this with total body mass, is actually higher for one of those groups. So in reality, the heavier mice have a lower metabolic rate. But if you divided that by body weight in grams, you underestimate the difference, because body weight, isometric body weight metabolic rate scales with an isometric body weight to the 0.75 power. That's the between species kind of analysis. Unfortunately, a thin mouse versus an obese mouse is not isometric. The obese mouse has much more fat, and the metabolic rate of adipose tissue is maybe a third that of lean body mass. So it's very difficult to try and correct metabolic rates in mice that differ by body weight. And on the left side is the example where you have mice that intrinsically have the same metabolic rate but differ in body weight. If you divide by body weight, now it looks like the heavier mice have a higher energy expenditure, and if you don't divide by body weight, you see the heavy mice look like they have a higher energy expenditure, and if you do, you overcorrect. So it's really a problem. So this is why I think everybody should always report for every calorimetry experiment the body weights of the groups. And the best way to really analyze and correct metabolic rate for body weight is analysis of covariance with both lean mass and fat mass as the independent predictors. Our problem is that almost all our experiments, we also measure body temperature, and the telemetry sensors are incompatible with the echo MRI measurement for body composition. But I just want to stress that this is done incorrectly and confusingly in many times, and this really can be done a lot better. Okay, so what I want to do now is get to the heart of the talk, which is how do we incorporate temperature into indirect calorimetry? And I'm going to talk about both ambient or environmental temperature, the same thing, I use them interchangeably, which is abbreviated TA. And I'm going to talk about core body temperature, which is abbreviated TB going forward. And the reason we have to consider this is that the mouse is really small, that if you look at the mouse, you see that it's much smaller than a human. And in fact, when you do this, you see that body weight-wise, a mouse is 3,000-fold smaller than a human. Despite being 3,000-fold smaller than a human, the median body temperature is the same diurnal rhythm, 1 degree warmer in the active phase. But because of the large body size of large mammals, the body core is insulated, whereas in small mammals, the body core is poorly insulated. Thus, large mammals get most of their heat from metabolic processes, and the heat is just a byproduct. It's not dedicated heat production to maintain body temperature, whereas in small mammals, they need metabolic heat production dedicated to increase body temperature, and they use brown adipose tissue for this. Now I'm going to just talk about thermogenic adipose tissue and brown adipose tissue interchangeably. That would also include beige adipose tissue. And at least for the experiments we're talking about, shivering could also, for example, be a contributor. But the main usual contributor is brown adipose tissue. Large mammals don't really use much brown adipose tissue usually. Now, the physiology in large mammals is oriented toward heat dissipation, very species dependent on how that's done. And the physiology in small mammals is oriented toward heat conservation and heat generation. And again, very species dependent on how it's done, but many behavioral adaptations, environment adaptations. We'll talk about huddling in mice later on. So in both large mammals and small mammals, body temperature is highly and tightly regulated. In large mammals, it's constant. In small mammals, it can vary in a regulated way, and I'll talk about some of those examples. So one of the things we've tried to do is incorporate the measurement of body temperature into understanding metabolic rate and physiology, and this just shows the kind of data we get. This is an example of data collection in one mouse where you can see on the top, after 2 days acclimation to the calorimetry chambers, they were put through 1 day at each of multiple temperatures from 22 up to 33 and then back down to 4 degrees, while continuously measuring total energy expenditure underneath that. Next line down is the respiratory exchange ratio, then physical activity, then core body temperature, and then food intake. And when you analyze these data, first just focusing on the body temperature, you see that the body temperature is regulated mainly by the dark versus light phase, that 1 degree difference, warmer in the active dark phase, but also by physical activity. Physical activity increases body temperature, and there's a slight reduction in body temperature with cold exposure also. Now, what I also want to do is review the way that we analyze metabolic rate data and its interaction with environmental temperature. And this is original, it's called the Scholander plot, initially published by Scholander, and references are up there. And what I'm doing is I'm giving it with examples of data that come from the same study on the last few slides. So what we've done here is graph the total energy, the resting energy expenditure as a function of ambient temperature. And you can see that the lowest temperature is the basal metabolic rate. The zone where the basal metabolic rate is constant is it's called the thermoneutral zone. The lower end of the thermoneutral zone is the lower critical temperature. And what this temperature is below this temperature, you can no longer maintain core body temperature with passive mechanisms to increase body temperature. Below it, you have to generate heat specifically to maintain body temperature, and the line is that measure of heat required. Now, if you extrapolate the heat required to 0, so you'd need no extra heat to defend core body temperature, that's the body temperature set point, or the defended body temperature. So you can measure the defended body temperature without ever measuring the core body temperature of the mouse. And this is a very useful, again, control for your experiment working the way you think it's working. And finally, the slope of this line is the heat conductance, which mathematically is the inverse of the insulation. So it's telling you how well or poorly insulated the animal is. So what we've done is take these data and sort of create the energy expenditure phase diagram for the mouse based on some assumptions and a lot of calculations. And you see that there's the basal metabolic rate in gray. At the top, there's the energy expenditure for physical activity. In the orange-brown color is the thermic effect of food, and in the blue color is the cold-induced thermogenesis. And we typically study mice at, say, 22 degrees, and you can see from this graph that at 22 degrees single-house mice in these conditions are expending 42% of their energy expenditure just to stay warm. So 42% of their food intake is being eaten just to maintain core body temperature. Now, that's really a big demand on the body, and that leads to some changes in biology that can occur if, for example, you fast a mouse. So this shows an example of the of torpor, which is an example where you take the mice in the first 12 hours in the light. Sorry, each red line is the core body temperature of an individual mouse. The light phase is the first 12 hours. The dark phase is the next 12 hours, shown in gray. And if you fast the mice for 24 hours, you see that 12 to 18 hours into the fast, they start dropping their core body temperature greatly, going into torpor. And torpor that requires only three things. It requires the inadequate food, as in fasting. It requires a cool environment. This is at room temperature. And it requires a quiet environment. If people are changing cages or doing experiments and making noise, the mice won't do this. This is a great weekend experiment. And so 2 points. One is this saves a huge amount of energy. And the second, this is normal biology for every mouse. All basically mice do this. This is not some weird, this is not a mouse dying. This is normal survival type biology. So we want to avoid this sort of pressure on the mice to do all this cold-induced thermogenesis. So what would happen if we study mice at closer to thermoneutrality, say at 30 degrees, and as a better model for human biology? So you can see we've been talking about mice at 22 degrees. If you now move them to a thermoneutral where we set cold-induced thermogenesis to 0, you now see that the components of energy expenditure in the mouse now much better match the components of energy expenditure in a typical non-over-exercising human. And that leads to the question, could studying mice at 30 degrees as an approach to thermoneutrality be more predictive of the efficacy of human drugs? And I'll just show you one example that we did with a model compound, which is dinitrophenol, which is a chemical uncoupler. And so what it does is it makes every mitochondria in the body work a little bit more and expend a little bit more energy. And so what you would predict at 22 degrees is that you would see a reduction in the brown fat function, and that's shown in blue as the energy expenditure caused by DNP, and that would be compensated for by a reduction in adapted or cold-induced thermogenesis. So overall, there's no change in total energy expenditure, food intake, or body weight or adiposity, and that's exactly what was observed. These are real data showing you. Now, on the other hand, if you study at 30 degrees, the prediction is you're going to reduce the adaptive or cold-induced thermogenesis and be able to see the drug effect. And again, that's exactly what we see. These are real data. There turned out at 30 degrees, there was a little bit of brown fat activity that was reduced. The total energy expenditure was increased as expected. However, somewhat to our surprise, the food intake didn't compensatorily increase to compensate for the increased energy expenditure, which we don't understand, but which is not the topic for today's seminar. So the point I want to make here is that how you study a mouse really matters, whether you're studying it at room temperature or at thermoneutrality or approaching thermoneutrality. You can get very different results. And the other point I want to make is that mice adapt to the ambient temperature. It takes about 1 to 2 weeks of cold exposure for full phenotypic browning or beiging of adipose tissue, and a similar time to involute the tissue with warm exposure. So you can make a mouse very cold intolerant by chronic exposure to, say, 30 degrees. And these warm adapted mice will not tolerate the cold exposure, but can tolerate it if they're gradually readapted to the cold. Even UCP1 knockout mice can tolerate cold just fine if gradually adapted to it. And finally, the central body temperature regulation adapts more quickly than the tissue changes, and we've shown recently that it probably is complete within 3 to 5 days, going from room temperature to 30 degrees or 30 degrees back to room temperature. And this, I just want to also give some technical things on how we assay for bad activation. So if your goal is to assay how functional is brown adipose tissue, then you want to be sure that the mouse have been housed chronically below thermoneutrality, so the bout is active. But then you acutely move the mice to 30 degrees to reduce the tone to the brown adipose tissue, and you get a basal 30-degree metabolic rate, and then you give drug acutely. And we do this all the time in the lab, and examples of drugs we've given are beta-adrenergic agonists, melanocortin agonists, BRS3 agonists, and if you give CL316-243, a beta-3 agonist, you end up with a doubling or slightly over a doubling of metabolic rate under these conditions for the assay. The next topic I wanted to talk about was the effective housing. Now, mice in the wild live one male, multiple females, multiple immature mice per group, and in the lab we never use those conditions. We don't want mice reproducing ridiculously, so we don't keep them in family groups. And when we want to study metabolic rate and body temperature and food intake, we have to use individually housed mice. Now, individual housing precludes all sorts of social behaviors, including huddling and fighting. The animal care guidelines suggest not doing single housing, but it's really unclear if single housing is more stressful, which it may be for females, but for males who are fighting, I think group housing may actually be more stressful. But be that as it may, we actually wanted to study the effect of group versus single housing, and did so with 3 mice versus 1 mouse per cage. And we summarized these results here. So basically, compared to 3 mice per cage, single housed mice had increased energy expenditure, whether they were male or female, to make up for the loss of huddling. However, it was kind of interesting that the females completely defended their prior body temperature by really increasing their metabolic rate, whereas the males let their body temperature drop a little bit so they didn't have to increase their metabolic rate as much. So the males are losing a little bit less heat than the females, which is kind of interesting, not something we had expected going into it. The other thing we studied that I want to talk about today is the sex difference in adaptation to fasting. So I showed you that mice can go into torpor and reduce their body temperature with fasting, and males and females do that. But the way they do that is different in that the males, when singly housed, increase their torpor episode number with no change in duration, whereas the females had no change in torpor episode number, but increased the torpor episode duration. So to summarize the social thermoregulation vignette, single housing increases heat loss and amplifies the effect of fasting in a cold environment. Male and female mice use different thermoregulatory strategies to respond to single housing, and single housing is more sensitive than group housing for studying thermal physiology phenotypes. For example, the huddling that group housed mice do totally precludes the kind of Scholander analysis I showed you that's so useful for understanding the components of energy expenditure. And finally, what I want to do now is spend a little bit of time on the upper end of the thermoneutral zone. And I'm going to introduce it with a slightly different graph, which is again a Scholander plot, but in this case for energy consumption per household in a city. And you see that just like mammals, houses and energy consumption, which is again the surrogate for the people living in the houses, shows the same sort of behavior, meaning below the lower critical temperature, you turn on your heat, it spends a lot of energy to keep the place warm. And then there's a thermoneutral zone where you're using passive things like opening or closing windows more or less closed to regulate your thermal environment. And then above a certain point, the upper critical temperature, you turn on the air conditioning, and that's energetically expensive, and you expend energy to reduce the environmental temperature. Well, we wanted to study the upper end of the thermoneutral, of the energy expenditure curve. And indeed, this is the way the curve is always drawn for mice also. However, when we looked to see if anybody had incorporated body temperature into studies of the upper end of the mouse thermoneutral zone, we could find none. So we decided to study it. And we had a very big surprise. I'm showing you first the mouse dark phase, or the active phase. And you see, if you look at the black line, that as you get warmer, the mouse energy expenditure minimum is about 33 degrees. And immediately after that minimum, energy expenditure starts increasing. So this says that the thermoneutral point for the mouse in the thermoneutral zone for the mouse in the dark is a thermoneutral point, which we'll call the thermoneutral point dark. Interestingly, what happens to body temperature is constant at the regulated dark phase temperature until you get to the TMP, in which case body temperature increases. So this says that once you get to the TMPD, above that point, the mice have lost thermoregulatory ability. Body temperature is increasing at almost a degree of body temperature per degree of ambient temperature. The mice can't thermoregulate above this temperature. Now we look at the light phase, and what we find is something very different, which is you now see the more standard Scholander curve for mouse during the light phase, where you do have this flat energy expenditure area. But if you look at the body temperature, you see at what we're calling the thermoneutral point for the light phase, there is, as soon as the energy expenditure starts to plateau, you see an increase in body temperature, which then increases in a biphasic way. And if you actually superimpose the resting and active periods, the light and dark phases, you get this, where you see that the mice in the light phase also lose thermoregulated control at exactly the same point as they did in the dark phase. So above the TNP dark, whether you're in the light phase or the dark phase, you see an increase in body temperature, you see an increase in metabolic rate, which is probably a Q10 effect. And so what I want to point out about this is that humans don't have this behavior. Humans have a thermoneutral zone that's, say, 10 degrees when studied in the active phase. So this is a fundamental difference in thermal biology between mice and humans. So what are the implications of what we've just shown you on the thermoneutral point for mice? So people always want, we for sure have been champions of this, we always want to study mice at thermoneutrality. But I'd like to argue now that studying mouse, quote, at thermoneutrality, quote, is not really possible or feasible. Having a thermoneutral point in the light phase of 29 degrees and a thermoneutral point in the dark phase of 33 degrees is not a thermoneutral zone of 29 to 33. Taking advantage of thermoneutrality, you'd have to cycle every 12 hours between 29 and 33 degrees, which is really getting kind of hard to do if you want to study mouse biology in our usual animal facilities. So how should one choose a mouse experiment ambient temperature that's most likely to predict human physiology? And there's a lot of there's a conflict in the literature between the Speakman and Cannon-Nettegard groups, where Kaye and Speakman have suggested you should study mice at 25 to 27 based on a percentage of BMR being analogous to humans. And Cannon and Nettegard suggest 28 to 30 because they say you really want to be at thermoneutrality. And we propose a TA of 28 to 29 for totally different reasons. We propose that you want to stay below the thermoneutral point at all times, including the light phase, because this would allow the mouse to use its thermoregulation, because mice typically live below thermoneutrality, and you don't want to be forcing them into an area where they're doing funky things. However, you want to stay near the thermoneutral points because you really want to minimize cold-induced thermogenesis, which is minimal in humans. And then I would also make a proposal that we state the actual ambient temperature used in our experiments and avoid using the word thermoneutrality, because I think it's very hard to achieve thermoneutrality in a mouse. So what are the implications of this? So many prior studies use 30 degrees, including ours, but 30 degrees is above thermoneutrality in the light phase. So how do we interpret this? I would argue that increasing the body temperature above increases the inflammatory immune actions, and therefore we may, by studying mice at 30 degrees, may be studying slightly activated or stimulated immune systems. And the point I just really want to stress, and I don't go through other examples, is that mice are not just small humans when it comes to thermal biology. They're not just quantitatively different, they have qualitatively different thermal biology. And with that, I'd like to summarize. Mice are typically studied at room temperature, well below thermoneutrality, while humans usually live and are studied at thermoneutrality. Drugs can have different effects when studied at thermoneutrality versus below it. The mouse thermoneutral zone is a thermoneutral point that changes diurnally by about 4 degrees. We propose considering body temperature in the definition of thermoneutrality. Currently, it is not. And I would argue what mice have what we've called thermoneutral points, not a thermoneutral zone, which must be incorporated into mice, are used to model human physiology, and that the thermal physiology of humans as mice is qualitatively different. And with that, I would like to thank the people that contribute. The current members of my lab are listed on the left. I'd specifically like to point out Wojtek Skop, who has done the TNP work and the huddling work, and Chunying Zhao, who's been a long-term collaborator on, I think, on all the studies I've shown. Oksana Gavrilova is even a longer-term colleague and collaborator who's an equal intellectual contributor to all the work you've seen. And I think with that, I will move on. And Oksana, who really has the answers, and I will be happy to answer any questions. I hope that was informative and useful. Thank you for the opportunity to give this talk.
Thank you so much, Marc, for that fantastic presentation. Hello, everyone, again. I hope that you found this presentation insightful so far, and I'd like to now bring back onto the stage both Marc and Oksana, and we'll now begin the Q&A session where we can get to some of the answers to your questions. As a reminder, if you have a question that you'd like to ask the presenters, you can submit it using the Ask a Question panel beside the media player. All right. Marc, Oksana, thanks for being here once again. I'm going to jump right into these first few questions that are coming in here. So this one is directed to Marc to start. And the question is TNP/TNZ similar in different strains of mice?
Darius, thanks for the question. So we have found that at least as we've measured it, the TNPs were the same in males, females, 129, black 6, diet-induced obese versus chow-fed mice. The only time we found a real difference in the TNP was or one of the only times was in OB-OB mice. And OB-OB mice think they're starving. So what they do is they behave appropriately for that. So they defend a degree lower body temperature, and that lowers the TNPs by about 2 degrees. So the quick answer is the TNP concept seems very constant, at least how we've measured it in our mice. The one example of mice where the TNP concept didn't really work was in mice that had neonatal ablation of the TRPV1 receptors that inform the brain of what the environment and body temperatures are. And in that case, the mice are sort of poikilotherm, so you can't determine the TNP, because in a cold temperature, the body temperature is colder than it should be, and in the warm, it's warmer than it should be, and you just get very different thermal biology. So in normal mice, we really have seen it very well behaved.
Excellent. Thank you so much for that answer. Hello, Oksana. Are you with us here?
Yes. Hello.
Awesome. This question is directed to you. How does the home cage environment with bedding versus a center feeder cage with plastic floor impact energy expenditure?
So as Marc has already mentioned, we use two different types of indirect calorimetry system in the lab, both from Columbus Instruments. The older model uses smaller chambers with plastic floors, no bedding, and with the feeder located in the middle of the floor. We have been using these type of cages for many years, and they worked very well for us. However, we've always had a concern that the lack of bedding and rather unnatural environment may affect thermogenesis and food intake. So when the new system became available, which uses bigger cages with bedding and basket feeders, which are very similar to normal mouse home cages, we decided to compare directly the performance of these chambers. And we used the same set of mice and subjected this in two systems. And we subjected these mice to a wide range of temperatures and calculated all parameters Marc just talked about. And I was pleasantly surprised that essentially every parameter we measured or calculated using these two systems were very, very close and agreed remarkably well. So I feel very comfortable using smaller cages after this experiment. We use both systems for slightly different types of experiment. For long-term experiments, we use home cage environment. For acute pharmacological systems, I prefer smaller cages. And you're most welcome to ask me questions directly if you have any technical, more questions about these two systems.
Great, thank you, Oksana. Another question for you. How does the TNZ/TNP depend on the assay conditions?
This is a very good question. Marc has already addressed diurnal variation of thermoneutral point. The other parameters that can affect the results are housing conditions. In our home cage environment, we use wood chip bedding, which, as I mentioned, had really minimal effect on measured parameters. And thermoneutral zone was the same regarding what chamber we used. But you can imagine that if you use a more fluffy bedding or nesting material in the cage, you may get different responses in the colder range and in the hotter range. So I would be cautious about using the bedding. Another parameter that can affect the results is the time at individual temperatures. The mouse spends at individual temperatures. We used two variations of the protocol. In the earlier studies, we subjected mice for 24 hours to different temperatures. And this is the graph, one of the initial graphs Marc showed. Later, we used a shorter window from 3 to 4 hours. And the results, when we compared these protocols, the thermoneutral zone was very similar. But again, we need to keep in mind that exposing mice, for example, to extremely cold or hot temperatures is stressful, it changes their behavior. And you might be measuring not acute response to temperature, but a combination of acute and semi-chronic effect. The other thing that we try to avoid is changing temperatures during the transition window from light to dark phase, because during that time mice are more active and temperature increases simultaneously with activity, so it may affect the shape of your the calculated parameters.
Awesome. Thank you so much, Oksana. Mark, I have a question for you. Chris Gordon showed mice will choose a cooler spot in dark in a thermal gradient. I think they choose 25 degrees Celsius. How does this relate to your suggestion to house mice at 28 to 29 degrees Celsius?
Yeah, so I think that the 28 to 29 answer is, how do you make mice more similar to humans? And so, Chris's thermal preference shows that mice don't want to be that high, but at least at 28 to 29, they're not going to be changing their body temperature, and they're going to be doing less cold-induced thermogenesis. So I think the ideal way to study mice is probably at multiple temperatures. Maybe pick two that say 29 or even 30, and then study them again at 22. But that just gets expensive. So that's my attempt at selecting, based on our data, what a temperature is that would minimize the cold-induced thermogenesis, but still allow the mouse to control its body temperature where it wants to. It's a good question.
Great.
And I think
Another
Steve
one
asked
for you.
another question, whether the TMP of the mouse changed with age. And we haven't studied that extensively, but we did not see it between the various groups we did study.
Great. Perfect. Thank you, Mark. We'll keep going with another question for you. Do you think differences in light and dark TNP is related to differences in RER, like using more fat during the light cycle?
No, I think that the difference in TNP is the fundamental regulation of body temperature. I think the main driver is that the mouse is trying to defend its day body temperature, its light phase or dark phase body temperature. And what happens in the light phase is it can no longer defend it that low. So it has this 4-degree regulated period where it, in a regulated way, arouses it to rise until it hits the TNP dark, where it can't defend it anymore. I think the RER is just telling you what fuels the mouse is using to achieve the energy expenditure that it needs to defend the body temperature. If I understood the question, and again, if anybody has questions that don't get answered here, I'm happy to answer them either through the Inside Scientific process, or you can just email me also. Happy to answer any questions individually.
Absolutely. We'll keep the questions rolling here. We still have a couple of minutes to answer a few. There are a lot of great questions that are rolling in right now. So, Mark, do you agree that if the RER increases when it indicates increased DNL?
Yeah, so if you fast a mouse, and if you put a mouse in a calorimetry chamber, fast them, the RER will drop, say, to 0.75 or something close to that. And then when you give back food, especially if it's chow that has a lot of carbohydrate, the RER will increase over 1, because what you're doing is doing de novo lipogenesis from the carbohydrate in the food to replenish your fat stores, and the RER will be over 1. So the point I was trying to make, that the RER and the food quotient should be similar, is when you're not, when you're not doing funky things like fasting a mouse, when you don't have lactation, when you don't have growth, those are the sort of conditions that can affect the net energy balance.
Great, thank you. Here's a question related to changes in humidity. Will the TNP change when humidity changes? Will the mouse's temperature sensations change?
Ooh, that's a great question. So somewhat to our surprise, it turned out that when we did our multiple housing, the humidity went very high. And so I tried to read about what's known about evaporative heat loss. So the only way you can reduce body temperature of the mechanisms, conduction, convection, radiation, or evaporation, that evaporation is the only way of getting rid of heat that will allow you to do it when the environmental temperature is greater than the body temperature. Now, it looks like if you look in the literature, mice do really not do much evaporative heat loss. They can do stuff like spread their drinking water over their fur, and they do these sort of, some of the mice managed to figure that out and did that. But the bottom line is mice are not really good at evaporative heat loss. Mice aren't used to needing to get rid of heat. They're more used to conserving it. So I think evaporative heat loss is minor, and I think therefore the relative humidity matters less for mice than it would for species that really do use a lot of evaporative heat loss. But that's mostly speculation on my part. We haven't directly quantitatively investigated that.
Amazing. Marc, do you think that mice housed at thermoneutrality are truly physiologically humanized?
I would argue probably not, which hurts me to say it because I've spent the last few years thinking that they probably were. But I think that mice are not just small humans, and even if you could do that magic switching of every 12 hours, staying at the thermoneutral point, even then I'm sure there's some biology we don't quite understand.
Great talk. Thank you so much, Professor Reitman. Are you aware of studies that, besides the one shown for DNP, have investigated the role of ambient temperature, 22 versus 30 degrees Celsius, for example, on drug-induced efficacy on weight loss?
Yes. We've done a couple of studies, but I didn't include here. One is we used the beta-3 adrenergic agonist CL316-243, and again showed a difference depending upon the environmental temperature. And but I think, the bottom line is inducing brown fat or putting a mouse in the cold is not a great way to lose weight, because a self-respecting mouse should just increase their food intake to match the increased energy expenditure. And so I think anytime you're talking about activating brown fat or using cold as a weight loss technique, it's going to have to be coupled with something that disrupts that normal coupling between increased energy expenditure due to defending body temperature and food intake. So I think the data are increasingly showing that activation of brown fat is better for glucose homeostasis than it is for body weight homeostasis. And I think if you did want to study it for body weight homeostasis, you're going to really have to work on combining it with some agent that inhibits food intake in some way.
Amazing. Here's another question for you, Marc. This one says, Marc, some of us rebels feel that indirect calorimetry is fundamentally flawed. For example, if the oxygen to ATP assumptions are incorrect. Might this explain the surprising TP, TNP, TNZ relationship that you found?
So I think everything we're talking about in our indirect calorimetry is we're not comparing to an absolute scale. We're comparing to what the mouse does at a slightly different temperature. So I think even if you feel the assumptions are wrong, that they're consistent through the experiments. And I don't think the assumptions underlying indirect calorimetry are particularly worrisome for any of the stuff that I've shown today. I think what we've shown is valid. Sorry.
Fantastic. Thank you so much, Marc. And of course, Oksana, for this great discussion. It was really fantastic to have you both talk to us about what you studied.
It was a pleasure. And again, both Oksana and I are happy to answer questions emailed to us or to Inside Scientific.